We know that plants capture sunlight to produce energy-rich organic compounds through photosynthesis. But photosynthesis isn't just one reaction. It is a two-stage process. The light-dependent reactions, there should actually be an S here because there are many reactions occurring in this phase. The first phase is the light-dependent reactions, and this entails the harvesting of light energy to make ATP and NADPH. The light-independent reactions, which is the second phase, also known as the Calvin cycle, uses these products to make sugars. However, for this video, we will focus on the light-dependent reactions, where sunlight splits water, releases oxygen, and generates energy carriers, which are ATP and NADPH. These reactions occur in the thylakoid membrane. You can imagine thylakoids as flat pancakes, and stacks of thylakoids are called grana, and one stack is called a granum. So this is the plural, grana. Let's zoom into the thylakoid membrane. The thylakoid membrane separates two spaces, namely the chloroplast stroma and the thylakoid lumen. So the lumen is the inside of the thylakoid and the stroma is the fluid outside. So the light-dependent reactions are essentially an electron transport chain that is powered by sunlight. The process starts when pigment molecules or chlorophyll in photosystem 2 absorbs light. Here's how that happens. Firstly, light harvesting complexes, which consist of proteins bound to chlorophyll molecules. So they are just proteins plus pigment. So these light harvesting complexes, they absorb the light energy, they capture photons, and they funnel or they transport that energy to P680. Now P680 is a special pair of chlorophyll molecules in photosystem 2. And it is called P680 because it absorbs light best at, you guessed it, 680 nanometers. So once P680 absorbs this light, the electrons in its structure become excited. And one of the electrons are ejected, and this leaves P680 with a positive charge. So now we got P680 with a positive charge, and this has become an oxidant. Now you may ask yourself, alright, P680 has ejected an electron. It has lost an electron and it now has a positive charge. Will it ever replace that electron? Yes, it will, and it gets that from splitting a water molecule. P680 splits a water molecule into oxygen, and this is the oxygen that we breathe, so it is released into the environment. And we also get protons, which will then contribute to the proton gradient, which is then essential for the production of ATP at the end of the electron transport chain. And we also get 2 electrons. And then P680 will replace its ejected electron with the electron from water. Now back to this excited electron. This excited electron is passed to plastoquinone, in short for PQ. And once it receives 2 electrons, so 2 electrons, it is reduced to plastoquinol, or PQH2. And PQH2 at the same time also takes 2 protons from the stroma, from the fluid in the chloroplast, the outside of the thylakoid. And it will take that and it will make its way to the cytochrome B6F complex. And you can see that this arrow is going in a downward direction. That's because the electron gradually loses energy because it is providing energy to plastoquinol to take the protons from the stroma and to make its way to the cytochrome B6F complex. So once plastoquinol has arrived at the cytochrome B6F complex, the electrons that it is carrying now is low in energy. So you can see the cat electrons here are quite tired. Plastoquinol, the cytochrome B6F complex will oxidize plastoquinol, so it will take these electrons away, and then the protons will then be transferred to the thylakoid lumen through cytochrome B6F complex. You can tell by now that the number of protons in the lumen is just gradually increasing, and the number of protons in the stroma is gradually decreasing. So this is how it will look like. Plastoquinol is donating, or is being oxidized by the complex and is giving up its electrons, and the protons are delivered into the lumen. So once plastoquinol gives up its electrons to the cytochrome B6F complex, it is oxidized back into plastoquinone, and plastoquinone shuttles back to photosystem 2 to receive another pair of electrons. From here on out, the low energy electrons in cytochrome B6F complex is transferred to plastocyanin one by one, by the way, and plastocyanin moves to photosystem 1. So again, plastocyanin takes the electrons from cytochrome B6F complex one by one to photosystem 1. Since, as the name states, the light-dependent reaction needs light, so it has to occur in the day. There's abundant light energy, and this light energy, just like how it works in photosystem 2, it hits the light harvesting complexes, and these light harvesting complexes, they funnel the energy to the special pair. Now, the special pair in photosystem 1 is different than the special pair in photosystem 2. In photosystem 1, it's called P700. And as you may guess, it's called 700 because it absorbs light best at 700 nanometers. Once it is absorbed, this light energy, it ejects an electron, and once it ejects an electron, P700 requires a new electron to replace the one that it has just lost. And this electron is the one that is transported by plastocyanin. So, recap. P700 gets the light energy, the photon energy. One of its electrons gets excited and shoots out. P700 now needs a new electron, and plastocyanin delivers. Plastocyanin delivers that missing electron to P700. And then plastocyanin just goes back to get that electron, and then P700 ejects another electron, and then the missing electron will be replaced by this one. So it's just a continuous process. After that, the electron is transferred to ferredoxin, one by one again. Ferredoxin has a job then to transfer these electrons one by one to ferredoxin NADP+ reductase, or FNR for short. FNR then uses two electrons to these high energy electrons to reduce NADP+ to NADPH, and this NADPH will then be used in, can you guess it? Used in the Calvin cycle, or in the light independent reactions that will take place in the stroma. And by now you may have noticed that the proton gradient in the lumen is gradually increasing, because throughout the entire electron transport chain, protons are just being transferred from the stroma to the lumen. And this, as I mentioned before, the proton gradient is extremely important for the synthesis of ATP. ATP is synthesized with the help of ATP synthase. This is a protein complex that consists of various subunits. You can think of ATP synthase as a molecular turbine or molecular motor that just spins and spins and spins. So when protons enter ATP synthase, this will cause the subunit, the F0 subunit, to rotate, and this rotation will cause a conformational change in the F1 subunit, and this allows ADP to bind with inorganic phosphate, which results in the synthesis of ATP. And ATP will be used, just like NADPH, in the Calvin cycle. So 1 ATP is produced per 1 proton. This is the general overview of the light-dependent reactions. I hope you guys enjoyed this video. If you have any doubts, if you have any questions, please do not hesitate to ask in the comments, and I will be glad, very glad, to answer your questions. I hope you all have a great day or a great night, wherever you are, and just like always, happy learning.